We now move into our final session, and then final session is advantage of third generation sequencing in PGT, insights from product and operational perspectives. So this session we will focus on how TGS is reshaping both clinical application and laboratory operation. So our first speaker in this session is Dr. David Wong Chen-En, the Director of Yikon Genomics Malaysia. So he completed his postdoctoral training at Children's Hospital Los Angeles, affiliated with USC, and bring intensive expertise in NGS and Preimplantation genetic testing. So today he will share how third generation sequencing can enable decentralization, making advanced genomic testing more accessible to smaller centers and in-house laboratories. So Dr. David, please. Thank you.
Hello. A very good afternoon to everyone, and thank you, team, for the kind introductions. It's my pleasure to be invited to PGT-A this year, and I will have the opportunity to share some of my experience as a lab from the lab user, as well as from the client's perspective, the application of our Yikon solution on the TGS product. So as we already have some quite number of the clients, especially in Southeast Asia, to enable them to use our solution on our PGT-based the TGS solution. So as today I'm going to the topic today I'm going to focus is on how we can leverage on the third generation sequencing technology to drive the transformation in the field of reproductive genetics. So I will give a brief introduction on the advantages of the third generation sequencing, and followed by today's main topic that will be include the PGT-A, as we call it as rapid aneuploidy screening solution, and also of course our advanced monogenic disease testing PGT-M, and also share some of our clinical data. So PGT technology has been evolved quickly, and currently we already in the 6th generations. And for the past decades, a lot has been focusing on the NGS, and is indeed the game changer with the very high throughput short sequencing rate capability, and it lower the price the cost per samples. But however, there is some limitation on this TGS that we are currently using the long sequencing rate to overcome this bottleneck. So these are some of the list of the advantages that could, I would say, to compensate some of the NGS limitation. But the main one would be the long sequencing read capability on the TGS, ranging from 15 to 20 KB long, or even longer, compared to this NGS solution. So because of this, I think it enabled the direct haplotyping without the problem, and offering the real-time result. So it overcomes some of the critical bottleneck that compared to your traditional NGS, and making the genetic testing more accessible and faster for the IVF patient. Of course, there's others list of the advantages on NGS. For example, you basically don't need the PCR amplicons to do it, and this will reduce some of the PCR bias. And of course, the equipment size itself is very, as you can see the picture here, is very small and portable compared to the traditional NGS. Is quite heavy and bulky. So basically, there are two devices option that fits every laboratory needs for the test. So they offer the entry-level solution, we call it MinION, and also the higher throughput solution, and that is, we call it as a Preimplantation Ion 2 Solo, or we call it P2 Solo in short. So for the entry-level MinION, it can generate the data around 20 to 30 gigabytes per flow cell, and it is very ideal for for a small lab or startup lab that with a low number of samples from the beginning. Whereas for the high throughput solution, P2 Solo, it can generate up to 100 GB per flow cell, and is ideal for the medium to high throughput number of samples. So, and both of our platform can integrate seamlessly with our Chrome analysis. And another I could share, one of our clients really likes this machine is because of low maintenance fees compared to a traditional NGS. Because you can see the machine is really small, and it does not have this kind of like very sensitive or expensive optics inside there, and the tubing inside there. So basically, you just need to like subscribe to the yearly warranty plan per year. So it will further lower down the cost at least like five times. So now we move on to our main topic today: how we apply the NGS on our PGT-A solution to our customer. So basically, there is no main difference in terms of the test content between the NGS and TGS platform. So, but there is significant improvement in terms of turnaround time and flexibility. So, why we call it as a rapid PGT-A? Because we have optimized the workflow and integrate perfectly with our WGA, either Chrome Swift, together with the library prep from the ONT, and of course with our Chrome data analysis systems. So this enables us to produce the data from sample to data in just 5 to 6 hours. So this will be useful for those with low or medium number sample and require for the fresh transfer. So from the lab perspective, I would recommend for the P2 Solo device for the more flexibility and high throughput sequencing, because you can later on expand to other applications, for example, the PGT-M, which I will talk in the next few slides. So not only for the PGT-A. So for P2 Solo, you have the two slot here. You can put two flow cell at one time, and one flow cell maximum, you can maximize up to 96 samples per flow cell, which means in the full capacity load, you can sequence up to 192 samples at a time. So for 96 sample flow cell, you require on average around 30 to 40 hours on average in order to get the 300K raw data per samples. So, and we recommending you can reuse the flow cell after that. So that means you can basically do 2 rounds on the same flow cell. This will further reduce the cost per samples. And if you have less samples, for example, you have 12 samples or less than that, basically you just need 1 hour in order to get the 300K raw reads. And you can reuse the flow cell up to 7 times basically. And our lab, yeah, as long as you have the enough number of pores inside there, you can just keep reusing the flow cell. So this is really provide the flexibility on in terms of the number of sample you sequence. So in the traditional NGS, you want to maximize the number of sample because of you cannot reuse the flow cell, right? So, but for in ONT platform, you can keep on reusing the flow cell as long as the flow cell is still in the good condition. So these are some of our validation data that we tested to compare with our NGS data. In total, 142 samples were tested, 98 from the clinical samples and 44 from the control sample. And we achieved up to 98% of concordance rate compared to the NGS platform, and up to 97.5% of sensitivity and 100% of specificity. So these are another set of performance validation data on the sequencing time with the different number of sample and number of pores. As you can see here, for example, with the 48 samples, or the over 6,000 pores of the flow cell, you can generate up to 80 GB or 150 million reads in just 72 sequencing time. But in a new scenario, you don't need actually like so many data for the 48 sample. As I mentioned earlier, you only need about 300K row reads per sample. So basically, for 48 samples, in total you might need about 15 to 16 million reads. So it will much lower down the sequencing time. And here again, we use the same number of samples. Here's 48 samples with the third time wash flow cell. We still can generate about 168 million reads number in total with the sequencing hour 51 hours. And we actually also just launched our PGT upgrade solution on the ONT platform. And now with this solution, we can check for the triploidy, UPT, and RH, as well as with the higher resolution at 4 MB. So this is this feature is particularly useful for the PN chat. And down here is our performance validation data. I think we achieved quite a good percentage of these in terms of sensitivity, specificity, and accuracy for triploidy and the UPD. And our TGS solution does not stop at the short sequencing read on the PGT-A because the unique feature on the TGS is able to do the ultra long reads, and that can advance our PGT-M solution with the direct haplotyping. So what is direct haplotyping and how our TGS-based PGT-M could really tackle on the clinical challenge here? So and one of the most common challenge in PGT-M is incomplete pedigree or lack of the proband. So even you have the proband, sometimes it's the newborn baby, and making the sampling is really difficult. And the second challenge will be the de novo mutation. So in the traditional method or NGS based PGT-M solution, you might need to have to sample the male sperm and female polar body, but this technique is really complex and time-consuming and laborious. Or you need to have the embryo haplotyping, or we call it as using the embryo as a reference. But in this case, you need to have enough embryo in that particular batch, and you need to have either the carrier or the affected embryos. Otherwise, you cannot like move on to the next stage of your PGT-M. But with all these challenges can easily solved by our TGS solution. We can directly obtain the SNP from the long reads data, or we call it direct haplotyping or phasing. So in summary, in the NGS platform, it requires complete family member for the pedigree analysis, at least 2 generations, as you can see here. Either you have the proband, or you get a sample from the grandparents. So in this case here, so if the couples are both carriers, you need to get both of the sample from each of their parents. So in total, you need to collect 6 samples or 6 blood. And with with TGS, we basically just need to collect the couple's embryo with any scenario. Either they both are carriers, we no need to without need any problem or complete pedigree. So before I go any further, I would like to just quickly go through what is the SNP linkage analysis and why the direct haplotyping is important in our PGT-M solution. So SNP linkage analysis is basically help to using the informative SNP to differentiate or distinguish the pathogenic haplotype from the normal one. So for example, this embryo, we can clearly know that this is inherited the maternal pathogenic haplotype, and the proband inherited both paternal and maternal pathogenic haplotype. And due to some of the limitation of the NGS with the short sequencing rate, as I mentioned earlier, it does not really help to link this SNP together. It's in a very small fragment size. Whereas for the TGS long reads, it produces about, you can read up to like actually more than 20 kb, and that can span all the multiple different SNPs on the same DNA molecule. And this enable our direct haplotyping construct from the carrier sample or the affected one alone without need of any problem. And currently we have two solutions on the PGT-M TGS platform. One is the 8-gene panel. It covers the common pathogenic variants. This is pure TGS solution. Or we have the NGS plus TGS combined solution. That is, we optimize for the more effective cost with the only with the affected sample on ONT. So these are the list of the genes that we cover on our 8 genes panel. It includes the most common one, the alpha and beta thalassemia, and we have the PKD1, SMN1, GJB2, hereditary deafness, and DMD and F8. So these 8 genes, from our clinical data, our in-house clinical data, this 8 gene panel actually cover 45% from our PGT-M cases from this, more than over 7,000 cases of our PGT-M. And so we move on a little bit more into a more technical things and what is direct haplotyping and how the direct haplotyping is done in our solution in the ONT platform. So we utilize this unique feature from ONT CoA adaptive sampling or as to get the to enrich the region of interest or region gene of interest. It's a PCR-free method. Instead, we use the software-based method to enrich the region of interest by we can uploading the back file and also the reference genome into the system. So what it does is that when during the sequencing, a single DNA molecule pass through this nanoprotein, and it will do the real-time base calling. And for the first few 100 base pair, it will align this sequence to the reference genome to determine whether or not it is in the region of interest or not in the region of interest. So if it is not, it will reject it, and it will, if it is the region of interest, it will continue to sequence it to produce the data. And how the actually this feature help to reduce the cost by at least 50 to 70%. So if you see, if without the adaptive sampling, you have to do the whole genome sequencing, and you can only cater for one sample per flow cell, and you get only like average 20 to 25 sequencing depth in the whole genome wide. But with the adaptive sampling, you can get about 5 to 10 folds enrichment of the region of interest, and more than actually in our lab we get like more than 30 times of sequencing depth in the region of interest. And per flow cell you can at least run 2 to 3 samples of this adaptive sampling per flow cell. So it will greatly help to reduce the sequencing cost per sample. So this is the overview workflow of our 8 genes panel on the pure TGS solution. So we can basically divide into 2 parts. There is direct haplotyping or the adaptive sampling parts, and also the embryo CMV and SMP part. So basically, we just need a couple of bloods and extract the high molecular weight gDNA and perform the adaptive sampling library or the SNP library for the 8 genes panel. And then we will perform, followed by the target sequencing on ONT for the mutation detection and SNP analysis. So for the embryo part, we'll perform the WGA and followed by the the CMV library prep and the SMP library prep. So all this data we can combine and then analyze using on our ChromeGo platform. So another solution, as mentioned earlier, is on the NGS plus TGS combined method. So for this method, we only perform the adaptive sampling of the carriers or the affected patient on TGS. And other samples of the normal one and the biopsy sample, we will run for the whole genome sequencing on the NGS platform. This is to reduce the cost per samples. And the beauty of our ChromeGo system is that our data analysis of the ChromeGo software can do almost everything. from the basic data analysis to a complicated one on combining the additive sampling data and PGT-M SNP linkage analysis. And as well as we provide this bed file, which is very important for the adaptive sampling. And you basically just need to key in the genes that you like to do for the adaptive sampling, and it will generate. You can download this bed file and upload to our Chromego system, and it works very well. Because we have optimized this bed file internally. And this is just another. On our Chromego, we also can do the adaptive sampling and SNP linkage analysis. It's very user-friendly software. You don't need to have the dedicated bioinformaticians to help you to do the analysis. You just follow the instruction with a few clicks of the button and upload the data for the analysis. And now, just want to share some of the case study that how we apply the TGS on our PGT-M test. So in this case, it's a de novo PKD1 mutation, and we have successfully construct the maternal pathogenic haplotype, highlighted in red color here. And from the SNP linkage analysis, the results show that all the 6 embryos is actually free from the carrier of the maternal pathogenic haplotype. So this data was further verified by using the Sanger sequencing, and this is the Sanger sequencing for the embryo, and indeed all the 6 embryos did not inherit the maternal pathogenic mutation. So another case that I would like to share is about the BRCA1 gene, and this is actually our first PGT-M ONT case in our lab in Malaysia. So this male has the heterozygous mutation on this BRCA1 genes, and the wife is normal. And actually these patients are from India. The international patient come to Malaysia for their IVF treatments. But when we asking them for the sample from the male's parents, they actually have the difficulties to get the sample, because it does not really make sense to ask the parents to fly all the way from India to Malaysia just to get the blood. So we offer them the ONT solution for them. So this is the phase 1 result. The male carrier was underwent the adaptive sampling and then pathogenic haplotype phasing. So these are the summary of the test result that shows we successfully detect the mutation, and we get a targeted N50 more than 38 kb, and with the coverage average coverage more than 33 times. And our Chromego actually also provide you the IGV view to show you the mutation point. And here is the SNP haplotype analysis result. The pathogenic haplotype is usually highlighted in the color, and you can see there's 2 colors here, and this is representing the upstream of this informative SNP, and the yellow one is representing the downstream of the informative SNP. In the phase 2, we got the CMV and SMP data for the couples and the embryo. So our Chromego actually provide additional information beside the SNP linkage analysis, such as the kinship analysis and the heteroploidy results, and also the origin of the aneuploidy, for example here. So our SNP linkage analysis show that this embryo, the number 6 embryo, is actually free from the paternal carrier of the paternal pathogenic haplotype, and these 2 carriers are obviously from the inherited the pathogenic haplotype. So in summary, TGS enable the ultra long reads that can help us to simultaneously detection of the diverse variants type, including the SNV deletion and insertion, CMV, and the structural variants. So, and it is applicable to all across the inheritance patterns, including the autosomal dominant, recessive, or X-linked disorder. And minimum sample requirement, as I mentioned, you only need the blood from the parents and not require any additional family member for the PGT-M. And with the high resolution mapping, it allows precise identify the breakpoints and the pathogenic loci. So, and this is especially including with the high repetitive or structurally complex genomic regions. And we continuously to expand our solution on the PGT-M platform. And for example, we are working on the carrier screening solution on the PGT-M platform, and as well as the ERT and EMT on the ONT platform as well. So, and then also we are expanding our genes panel for the PGT-M more than beyond 8 genes panel that I mentioned earlier. And with that, I just want to take this opportunity to kind of like show our lab. We just recently opened a reference lab in Malaysia, in the heart of Kuala Lumpur. And we welcome most of it if you come from within the region to visit us in Malaysia, and we can help you to further discuss how you can enable you to in-house our PGT-M solution by yourself, or you can send us for the servicing. With that, thank you very much, and open for the question to the floor.